So let me give the brief introduction about the Infosys Award and about Professor Shukla. So Infosys Prize 2023, laureate in life science, Arun Shukla is professor and Sonu Agrawal Memorial Chair, Biological Sciences and Bioengineering at the Indian Institute of Technology, Kanpur. Professor Shukla was awarded for his outstanding contributions to the biology of G protein coupled receptors, a key component of cell functions. His studies have elegantly illustrated mechanisms that selectively activate signaling pathways downstream of GPCRS. These and other discoveries provide fundamental understanding of the context-dependent function of GPCRs and suggest a new framework for the design of novel therapeutics. In his Infosys Prize Lecture today, Professor Shukla will talk about his research that envisions the design of novel drug against several human disease conditions with minimized side effects. The Infosys Prize Lecture Series is intended as a platform for the Infosys Prize laureates to speak about their award-winning research to a wider audience of research scholars and students. The lectures are meant to spark conversation and enable an exchange of ideas among the scholarly community and aspiring researchers. The Infosys Science Foundation, which gives the Infosys Prize, was set up in 2009. The Infosys Prize is an annual award to honor outstanding achievements of researchers and scientists in 6 categories: economics, engineering and computer science, humanities and social science, life sciences, mathematical sciences, and physical sciences. From 2024, the ISF is moving in a new direction and awarding early career researchers aged 40 years and below. The foundation works with communities of students, educators, and others to create conversation and around science research. The foundation is attempting to foster dialogue around science and society through talks, workshop, initiative, performance, and other events. Our events are a part of this larger objective to spark conversations and kindle curiosity about science and scientists. With these words, I invite Professor Shukla to deliver his talk. Thank you.
All right, it's a pleasure to be here. I remember that I came to BHU almost 20 years ago, that time to write the admission test to get admission in Bachelor's of Science BSc program. Must have been 20 plus years ago. After that, I visited this beautiful campus a few months back for an SERB program advisory committee meeting. And now this is my third trip for this particular lecture. It has been a pleasure every time I have come here. And despite not being directly associated with this wonderful institution, I feel connected in more than one ways. Your ex-Vice Chancellor, Professor Rakesh Bhatnagar, had taught me biochemistry when I was doing Masters in Biotechnology at JNU. Your current Vice Chancellor, Professor Sudhir Jain, was a faculty colleague at IIT Kanpur. And in our Department of Biological Sciences and Bioengineering, there are several faculty colleagues, at least a couple, who have done their PhDs from BHU. And more importantly, there have been several students in our PhD program, including several in our own lab, who have joined our programs after completing their masters from different departments in BHU. And those students are doing absolutely wonderful science, and therefore it's a pleasure to come to this place. And I can see that there are some students in the audience, and just so you know, I have come here primarily for you, so that I can share some of our science with you, and hopefully inspire you a little bit to continue to do wonderful stuff that you guys have been doing. So what I will do today, in the next 30-35 minutes or so, is give you an overview of what we do in the lab and how this might have an impact on designing better therapeutics. I understand that the audience can be quite diverse from different backgrounds. So I'll try to keep things very, very simple without getting into too much technical details. If it is too simple in some places for some of you, please bear with me. If it becomes a little technical in some places, I request you again to bear with me. But I'll just try to keep it as simple as possible. And if there are students who would like to interact after the lecture, I'll be more than happy to talk to you about science, about non-science, about anything that you want to discuss. So our lab at IIT Kanpur works on a class of receptors known as G-protein coupled receptors. And before starting with those receptors, let me just orient you a little bit in case there are people who do not have a biology background in the audience. As you know, most organisms, almost all organisms, they have cell as the building block, right? And cells have nucleus that contains the DNA, that is the genetic material. Cells have different organelles, Golgi bodies, endoplasmic reticulum, ribosomes, lysosomes. And these cells are surrounded by a membrane. So this is the outer covering that surrounds the membrane. And this is a lipid membrane bilayer. This basically serves as a barrier so that things cannot go in and out of the cell freely. It serves as a semi-permeable barrier, as some of you might have studied in your bachelor's or even before that. But cells have to communicate with the outside environment. All the cells which are present in our body, even if you talk about the cells in your skin, they are continuously exposed to a different kind of stimuli, different kind of signals, ligands, substances, and so on. And same is true for the cells which are present in different tissues. And therefore, the cells have to have a mechanism to be able to recognize the signal from outside and communicate that information to the inside of the cells. And for this purpose, they have several different types of protein molecules. Proteins are biological macromolecules. And these proteins which are embedded in the plasma membrane or other type of membranes as well. So the cells will have the outer plasma membrane, but even the intracellular organelles will have different type of membranes. And these proteins which are embedded in the membrane, they are known as integral membrane proteins. They are proteins, they are embedded in the membrane, and they are integral in the sense that they traverse the membrane bilayer. There are different types of membrane proteins. They can be ion channels, for example, potassium channel that will allow the permeation of potassium across the cell, sodium channels, chloride channels, and so on. The other class of membrane proteins are transporters that will allow the movement of different substrates or solutes, such as glucose transporter that will allow the transport of glucose across the cell membrane. And then there are receptors which do not physically allow the movement of anything across the membrane, but they allow the communication or transmission of the message from outside to inside. And these are known as receptors because they receive the signal. And the signal can be any chemical compound, a hormone, any environmental factor, and so on. And the receptors that, again, receptors are of different kinds. We work with one specific type of receptors in the lab, which are known as G-protein coupled receptors, GPCRs. They're also known as 7 transmembrane receptors, 7 TMRs, because they span the membrane 7 times. That is, these cylinders that you see here, these are membrane traversing helices which are present in a given GPCR, and they go in and out 7 times. And these receptors have their N-terminus. As you know, any protein will have N-terminus and C-terminus. These are the 2 ends. N-terminus is on the outside of the cell, while the C-terminus is inside. And these helices or cylinders are connected through extracellular loops, ECLs, or intracellular loops, ICLs. So these helices are connected on the outside and connected on the inside as well. These receptors, GPCRs, they constitute the largest family of receptors in the human genome. There are about 1,000 different GPCRs in our body, and they recognize a diverse set of stimuli. They can bind to and be activated by different hormones, peptides, small molecules, lipids, and so on. And pretty much there is nothing in our body that happens without a GPCR being involved. So the reason why you can look at the slide is you have a specific receptor in your retina, in your eye. When the photon of light hits that particular receptor in your retina, that receptor becomes active, and then the message goes to the brain. You can form the object, you can visualize things. That receptor, some of you might have heard about, is known as rhodopsin. That is the visual receptor. That is a GPCR. The way you can smell the perfume of the person sitting next to you, because in your nasal epithelium, you have olfactory receptors that bind to specific olfactory molecules and send the message to your brain so that you can tell if somebody is smelling nicely, if somebody's perfume you like or you do not like. If you drink coffee and you can tell if the coffee is bitter or sweet, that you can do because you have taste receptors in your tongue epithelium. So these are the receptors that sense the taste, send the message to the brain, and you can tell if the coffee is bitter or sweet, you need to add more sugar, it's too sweet, or whatever. And these taste receptors are also GPCRs. The way your heart is beating, the way your immune response is regulated, the way your blood pressure is regulated, the way you know that you are hungry, you have to eat something, or the way you know that you have eaten enough, you have to stop eating. All these processes are mediated by different receptors, different GPCRs. And therefore, it is very important to understand how these receptors work, not only from the point of view of fundamental signal transduction, but also to utilize that information to design, hopefully, one day better drugs or better medicines. So these receptors, because they're involved in so many different physiological processes, it is not surprising that about 40% of the medicines that are currently prescribed by the doctors, they work by targeting a GPCR. That is, they will either activate a receptor or inactivate a receptor, and that is how they give you therapeutic effect. And even if you look at any 5-year time period, you will see that most of the GPCR, most of the drugs, medicines, which are being approved and then they go to the clinic, they target GPCRs. This fraction of targetable GPCRs continues to be on the top. And there are different types of GPCRs, as I said, and this is just taken from a review article to show that different receptors, which are different families of GPCRs, continue to be targeted by different drugs, which are in, some of them are in phase 2, phase 3, and so on. So not only in terms of approved drugs, but even drugs those are being developed as of now. GPCRs continue to be one of the hot targets for these medicines. Some of the examples of GPCR targeting drugs are given here. If the fonts are too small to read from the back, I'm sorry about that. But some of you might have heard about asthma. And one of the drugs that is given in the inhalers, for example, is a beta-2 receptor long-acting agonist, salmeterol. Beta-2 receptor is a GPCR. People who undergo specific surgeries and have chronic pain to sort of manage their pain in terms of analgesic compounds, opioid receptor agonists are prescribed. For migraine, for example, serotonin receptor antagonists are given. Even in different cancers, even in mental disorders, neurological disorders, many of these receptors are the key targets for these medicines. Many of you might have taken anti-allergy medication at one point or the other. These anti-allergy medication, for example, Claritin, that goes and antagonizes or inactivates the histamine receptor in our body that sort of gives you relief from the allergy symptoms. Now, these receptors have been studied for several decades, and there has been a lot of research that has given rise to the current paradigm of signaling and regulation of these receptors. And this might get a little too technical, so I'll just try to simplify this for you. It is completely okay if you do not get the entire paradigm. There are just a couple of key points that will be helpful to know. So GPCRs are sitting in the membrane. When an agonist binds, agonist is a ligand or a signal or a stimulus that will activate the receptor. When the receptor becomes active, that is, it changes its structure or conformation, so to say, and then it interacts with another class of proteins which are present inside the cell, and these proteins are known as heterotrimeric G proteins. And because activated receptors interact with the G proteins, that is how they have gotten their names, G protein coupled receptors. Once G proteins are activated, they will give rise to different second messengers such as cyclic AMP, inositol phosphate, calcium, and we will get what is referred to as downstream signaling, activation of different signaling pathways, gene expression, cellular outcomes, and so on. Now, this signaling has to be regulated, because if the signaling continues to happen inside the cell, this will completely mess up the cellular homeostasis. The cells will just go airy. And to control this signaling response in spatiotemporal manner, cells have devised a mechanism that is known as GPCR desensitization. And essentially what happens is that activated receptors are phosphorylated. Phosphorylation is the attachment of a phosphate group to any target, in this case GPCRs. So receptors are phosphorylated, that is, they are attached to specific phosphate molecules. And then another protein that is present inside the cell that is known as beta arrestins, they come and interact with the receptor. Right? So receptor was activated, it was signaling through G proteins, now it has undergone phosphorylation, and now it is in complex with another protein known as beta arrestins. And it is believed that beta arrestins, once they are bound to the receptor, they basically preclude further G protein coupling because they occlude the binding site for G proteins on the receptor molecules. And that is how they are able to desensitize or stop G protein signaling, right? So this is the mechanism to downregulate or stop G protein signaling, if you will, in a very crude sort of manner. But over the last 2 decades or so, it turns out that beta arrestins have additional functions. That is, they can mediate traffic from the membrane to the inside in specific vesicles that are known as endosomal vesicles, endocytosis, that is receptor internalization. Receptor from the surface, that is plasma membrane, getting inside the cell. They can also mediate receptor ubiquitination and degradation. But most importantly, these beta arrestin molecules, they can also nucleate other signaling components, different kinases and phosphatases, and they can induce or initiate a parallel wave of signaling parallel to G proteins, right? So this whole thing is summarized in this slide. So unlike what is there in the textbook of biochemistry, these receptors can signal through two parallel pathways, that is G proteins and beta arrestins, right? So you can imagine that once the receptor is activated, G protein signaling starts, then beta arrestins come and bind to it, stop the G protein signaling, and start their own signaling that is referred to as beta arrestin signaling. Now, this is not very uncommon in signaling context, but there is something associated with this bifurcation of downstream signaling that makes it really, really exciting. And that is the cellular and physiological outcomes that are linked to G protein signaling versus beta arrestin signaling are quite distinct from each other. That is, when a receptor is activated, G proteins are activated, you will have cellular or physiological response 1. The same receptor, G protein signaling is shut down, beta arrestin signaling starts, you will have cellular and physiological response 2. So these are distinct physiological responses that can happen downstream of these signaling pathways. And the reason why this is interesting, this is a natural phenomenon, but we can modulate this in the lab by designing specific molecules that will activate either one arm of the signaling or the other arm of the signaling, right? And this is known as biased agonism. The terms are not so important. Essentially what it means is that it is possible to design a chemical molecule, for example, or a drug molecule that will bind to a given GPCR and tell the receptor that you have to activate pathway 1 or pathway 2. You have to give a specific response 1 or specific response 2, right? And this is basically summarized here. So as I mentioned earlier, when an agonist will bind, you will have both pathways activated, right? So agonist, you can imagine the natural hormone that activates a GPCR. Antagonists are molecules, these are synthetic molecules that will bind to the receptor and they will basically completely shut down the receptor. There is no G protein activation, there is no beta arrestin activation. But in the lab, we can design molecules which will bind to a given receptor and tell it that you have to signal through beta arrestin, or a different molecule that will bind to the same receptor and tell it that you have to signal through G proteins. And these ligands are known as either beta arrestin biased ligand. Why? Because they bias the signaling, or they favor the signaling towards beta arrestin over G proteins, or G protein biased ligands because they preferentially activate or favor the signaling through G proteins and not through beta arrestins, right? Now let me give you one example how this is exciting in the context of novel therapeutics. So some of you might have heard morphine, the word morphine, or you might have studied about this. This is a very powerful analgesic. It's a very powerful, strong painkiller. So after surgery, for example, you might have heard that patients are given morphine drips, right, just to subside the pain. And the way it works is that it's an agonist of a GPCR that is known as mu opioid receptor. Again, the name is not so important. And being an agonist, it will activate both the G protein pathway and beta arrestin pathway. So both arms of signaling are activated. It's a very powerful analgesic. It subsides the pain pretty strongly. But there is a problem. And the problem is that the patients who take it, they will have problems with respiration, they will have problems with constipation, they will have problems with addiction, tolerance, and so on. So the pain subsiding effect, that is the analgesic effect, is the desired effect. That is what you want to get. But because it has activated the receptor, you are going to get the side effects as well. It turns out that the beneficial effect or the desired effect of this drug goes through the G protein pathway, but the side effect that nobody wants, those go through the beta-arrestin pathway. So there is a bifurcation in terms of which signaling outcome is giving you the desired effect versus the other signaling pathway that is giving you the side effects. So now I guess you're starting to get the idea already that if you modify the structure of morphine in a way, or you design a new ligand that will be like morphine, that it will bind to the same receptor but selectively activate G protein pathway and not the beta-arrestin pathway, what you will have is the pain relief without the side effects, right? So now you can have a better version of morphine, a new drug, if you will, that will maintain the beneficial effects without giving you the side effects. Now, this used to be just a conceptual paradigm until recently, but back in 2020, a company known as Trevena in the United States, they developed a G protein biased ligand at this receptor, and then that is known as oliceridin. And because it was G protein biased, it was expected to have the analgesic effect without the side effects. It went through phase 1, 2, 3 clinical trials, and then it was approved by FDA, that is the Food and Drug Administration in the United States, as now a clinically prescribed drug. So this was the first GPCR biased drug that is a better version of morphine, if you will, to give you the pain relief, analgesic effect without the side effects. And when this entire concept sort of originated on selected receptors, it turned out that the same conceptual framework is applicable to almost every GPCR. That is, every receptor, every GPCR will have this bifurcated signaling. In almost every case, you have these two pathways giving rise to very different physiological outcomes. In some cases, it is the G protein pathway that will give you the good effect, and beta-arrestin pathway giving you the bad effect. In some cases, it is the other way around. That is, G protein pathway is giving you the bad effect, and beta-arrestin pathway is giving you the good effect. And this paradigm has now been established for almost the entire set of receptors that have been studied in different labs. And this has led to a complete shift in the drug discovery pipelines at major pharmaceutical industries. Now, every major drug discovery company has a program on GPCR biased agonism. Because if you think about it, they have a huge opportunity to design new drugs without having to discover new targets. In drug discovery, target discovery is also a very critical and very difficult part. But in this case, they are able to sort of use the same target receptors for which we already have the drugs and design biased drugs to improve the therapeutic window or efficacy over the existing ones. Now, the way it is done, including something that we have been involved in for the past several years, is something that is referred to as structure-guided drug discovery or ligand discovery. So for drug discovery, for ligand discovery, there are many different ways. The most classical or the original way has been that you screen a very large number of chemical compounds, and you hope and pray that some of those compounds will bind to your target, that is a GPCR, and then you can develop them further by modifying their chemical structures, they're reducing their toxicity, increasing their specificity, and so on. And that has been very time-intensive and effort-intensive process as well. And the newer paradigm is if you can really determine the structures of these targets, let's say GPCRs, you know how the given drug binds to the receptor, to a given receptor that is, and now use that structural information, that atomic structure of a drug binding to its target GPCR, and start from there. You just start to modify the chemical structure of those drug compounds and see, can you really encode biased ligands or biased agonism in those drugs, and then you can take them through different clinical trials, and hopefully some of them will pan out and become new class of drugs. So the idea is that if you start with a structure of a GPCR using different methods, X-ray crystallography or cryo-electron microscopy, and then using the structure, you can now screen very large chemical space. So when you, for example, screen chemical libraries, if you're doing it in the lab setting, even in cell culture system, you can screen a few million compounds. But if you do this in a computational, using a computational framework, we are able to now screen several billion compounds pretty quickly, within a few days, which means that you don't really have to even make all those compounds. You can simply explore the chemical space, theoretical chemical space, if you will, and make only those compounds which are likely to be targeting your receptor of interest. And once you have that, then you do a whole bunch of optimization, preclinical studies, and then different phase trials, hoping that some of them will survive and become drugs one day. Now, I'll just give you a couple of examples from our lab that we have been able to do in the past few years in terms of the structural visualization of these receptors. And now, using those structural templates, we are trying to design newer ligands, hoping that some of them will turn out to have better efficacy or therapeutic window one day. As I said, for any protein crystallography or protein structural determination or visualizing protein structures, there are different methods. You can use X-ray crystallography, you can use NMR spectroscopy, and a method that has become more popular recently is cryo-electron microscopy or cryo-EM. And I will not give you all the technical mumbo jumbo, but just to tell you very briefly, the idea is very simple, where we try to visualize individual molecules, let's say proteins or GPCRs, and simply try to calculate the structure, that is atomic structure of that given target protein or the receptor. So some of you might have used microscopy to visualize cells, right? That is at certain resolution. You can see the whole cells. If you use, for example, confocal microscopy, you can perhaps label the individual organelles, and so on and so forth. In this case, we are simply trying to visualize or image individual protein molecules, right? And the basic principle is very simple. You take your sample, you quickly freeze it at very low temperature, and then you put it under electron microscope. That is, you have an electron beam which you use to illuminate the sample, and then you can visualize these individual protein molecules. These are very, very tiny molecules, proteins or DNA, right? Unlike cells, if you will. And then if you basically average a large number of these individual molecules, if you can visualize them from different directions by doing some calculations, you will be able to reconstruct their three-dimensional shape. And this three-dimensional shape is basically the structure of the protein. So if you take a sample and simply put it under the microscope, it can have different biological macromolecules, proteins, for example, or something else. When you simply take an image, you'll not be able to make much sense of it, because you are looking at individual molecules in each of these squares. But if you merge a large number of individual molecules, perhaps you will start to see that some features emerge, right? So you average these millions and millions of molecules, and if you are basically looking at the sample from multiple different angles, you can reconstruct how the molecule looks like in a three-dimensional context. And that is basically your structure that you can then use for ligand discovery, for drug discovery, and so on. Now, this particular method, that is cryo-EM, has become more popular in recent times, because unlike X-ray crystallography, which involves growing three-dimensional crystals followed by diffraction of X-ray from those crystals. This method does not require any crystallization. You do not have to grow any crystals. You can simply take your proteins and put them under the microscope, and you can visualize them. It's more difficult than being that straightforward, but different protocols, different pipelines that have emerged, they have made it certainly tractable for many, many difficult and challenging proteins. And I'll just give you a couple of examples from the lab that, as I said, we have done in the past few years. So one of the receptors that we looked at is beta-1 adrenergic receptor. That is a key drug target for asthma. And in asthma, as some of you might know, you have this constriction of the blood vessel. And basically, in the inhaler, one of the key ingredients is long-acting beta agonist. That is an agonist that will activate the beta receptors, which leads to bronchodilation, and you get better breathing, if you will. And to visualize this, we took beta-1 receptor, we expressed and purified, that is, isolated it from the cellular membranes, and then mixed it with one of the asthma drugs, that is formoterol, and then visualized the structure using cryo-electron microscopy. And in this case, the receptor was bound to one of its interaction partners, that is beta-arrestins. So this is a raw micrograph showing the individual molecules of the beta receptor that is bound to formoterol and beta-arrestin as its interaction partner. So again, in this micrograph, there is not much you can tell. Water. But if you start to if you merge large enough number of particles, you will start to see some kind of shape in these molecules. And if you continue to do that, that is merge more and more individual particles and improve the signal to noise ratio, you will start to see these individual structures popping up. And by reconstruction, you will be able to determine the whole structure of this protein. This ribbon-like or wire-like structures are basically the beta-1 adrenergic receptor, that is the GPCR we are looking at. This is just one way of depicting the protein molecules in terms of their structure. Formoterol, shown here is in the pink. This is the asthma drug that we had included during this process to bind to the beta receptor, and that can be visualized here. And if you zoom in, that is, you looked at the atomic details, you can make out the chemical structure of this formoterol molecule and how this is sitting in the binding pocket of the beta adrenergic receptor. And now, using this information, it is possible to modify the structures in a way that you can generate biased agonism and then subsequently test the effect of those new compounds on different cells and then in mouse model and so on. The other example I'll give you is based on complement anaphylotoxin receptors. Again, these are just some GPCRs. The names are not so important, but their functions are. So when a pathogen infects our body, virus or bacteria, our immune system will become active and it will try to combat that pathogenic infection. And there are different mechanisms, antibody-based and macrogenetic complex. There is something which is part of the innate immunity known as the complement system. That is, there are several complement molecules, which are essentially small peptide molecules or even proteins, that are released by specific cells, and these complement molecules will bind to specific receptors, activate specific signaling cascade, leading to inflammation, and then subsequently, through different mechanism, clearance of the pathogens and so on. Those of you who are taking immunology as a course, perhaps you will study about this in your master's, perhaps. And these complement anaphylotoxins, which are very potent anaphylotoxins released by our immune systems to combat the pathogenic infection, they bind to different GPCRs. Again, these receptors are known as C3AR because the anaphylotoxin is C3A, or C5AR because the anaphylotoxin, that is the ligand, is C5A. And we have determined structures of these different complement receptors using cryo-EM. If you cannot read what is written there on the slide, that is completely okay. You don't really have to read. Basically, these are different atomic resolution structures of complement receptors in complex with different complement anaphylotoxin peptides or proteins that are released by our immune system. Some of these are human receptors, some of these are mouse receptors, and we have essentially visualized how the complement anaphylotoxins bind to and activate their corresponding GPCRs. I will not bore you with all the technical details, but I'll just give you a couple of slides to show one key point about these receptors. And these are two different receptors shown in different color. This thing in green is complement anaphylotoxin C3A. This thing in blue is complement anaphylotoxin C5A. They are binding to their individual receptors. That is, they are two different molecules. They bind to two different GPCRs. But if we sort of overlay them on top of each other, we see that the receptors look quite similar, but their ligands, they bind in a diagonally opposite manner, right? Which means that even if complement C3A toxin is released, it will bind only to its corresponding receptor, that is C3A, and not the C5AR, and vice versa. And this selectivity based on their binding pose is designed so that depending on specific pathogen context or depending on specific cellular context that has to be activated, one or the other receptor can be activated. So this is a selectivity that is built in the system by our immune system. Now, if we zoom in, we can see what part of these toxins or anaphylotoxins, they essentially interact with the receptors. So now we are simply looking at more zoomed in version of these structures, and we see that there is a thread-like structure at the end of this ligand, which is sitting in a specific binding cavity on the receptor, right? And now using this information, we can then change the structure of these ligands chemically or even design new chemical ligands. And in this case, I'm presenting two different agonists, one that has this interaction here in a small binding pocket on the receptor, and the other one that does not have this cryptic interaction on the smaller binding pocket on the receptor. And just by the small change in the structure of the ligand, we are able to encode specific functional response. That is, on the left side, we are looking at the G protein response for the C3A complement receptor. On the right side, we are looking at beta-arrestin response. So two different signaling pathways, right? And we see that agonist 1 can activate, agonist 1 and 2, they can both activate G protein response almost equally. This is a simple dose response curve measuring G protein activation. But agonist 2, which does not have the engagement with the small cryptic binding pocket on the receptor, is significantly weaker in terms of the beta-arrestin response compared to agonist 1 that is in blue. So by looking at these structures, we are able to modify these ligands or design ligands to have one function or one activation pathway intact while compromising on the other signaling pathway or the signal transduction pathway. And now we are testing these ligands in preclinical studies to see if we can take them further in terms of their beneficial and side effects. And finally, I'll just give you something that we have done very recently. Again, a different GPCR that is known as the Duffy antigen receptor for chemokines. This is an interesting receptor because it is expressed in our erythrocytes. Erythrocytes are red blood cells, RBCs. And as you know, if somebody is infected by malaria, the malarial parasite Plasmodium vivax enters the erythrocytes or RBCs. You might have studied about the entire Plasmodium life cycle. And this invasion of RBCs by Plasmodium parasite is a key step. And this particular receptor that is expressed at the surface of these RBCs serves as the anchor point or binding site for the Plasmodium vivax. Right? So this receptor is sitting at the membrane to which the Plasmodium interacts and then subsequently invades the cells. It also serves as the anchor site for something called pore-forming toxins, which are specific toxins secreted by different pathogenic bacteria such as Staphylococcus aureus. And we have recently determined the structure of this Duffy antigen receptor in complex with its chemokine ligand using cryo-EM. And this has allowed us to visualize, for example, what is the binding interface that is utilized by Plasmodium vivax or Staph aureus through its pore-forming toxins. And now we are designing molecules to inhibit the interaction of Plasmodium or even Staph aureus toxins with this particular receptors as a way to potentially prevent host pathogen interaction. So essentially, I have given you a brief overview of how structure-guided approach can be utilized to visualize the binding of different ligands or drugs to the receptor, and how we can use this information to design, hopefully, better therapeutics and minimize the side effects. Those of you who are more interested in the technical details, I'll be happy to chat with you. I try to keep things very simple. I know still it got a little technical in some places, but if you have specific questions, I'll be very happy to chat with you. These are the people who have done all the work, and it is my true pleasure and honor to have such wonderful students and fellows in the lab. I'm standing here today in front of you because of all their hard work and effort. I primarily, as I always say, sit in the office and drink a lot of coffee. They are the true superstars in the lab. And I just cannot imagine, without them, we'll not be able to sort of, do whatever little we have been able to do over the past several years. We also have a very extensive global collaborative network because biology, as some of you would know, is very complex, it's complicated, and one lab cannot become an expert in every technique or every method. So we collaborate with a lot of people across the globe who bring different complementary expertise on the table, and then working together, we are able to address some of these key questions in GPCR biology in a holistic manner by applying multiple approaches in parallel. We are supported by DBT India Alliance, DST, IIT Kanpur, of course, CSIR, and we are very, very thankful to our funding agencies. They have basically believed in us and supported us to do our research. I just wanted to show this image quickly. When I started in India almost 10 years ago, that is 2014, we had 0 cryo-EM facilities in the country, that is high-end cryo-EM facilities. And this is something which did not sit very well with me. So obviously this has been a collective and collaborative effort of scientists, policy bodies, policy makers, and different funding agencies. But we have been able to approach them and to convince them that this is a direction where as a country we must invest in.
And now the scenario has changed. We have about 10 different high-end cryo-EM facilities in the country, including one at IIT Kanpur. And these are the same setup that you will get at any other foreign institution. So unlike synchrotron, that is for very high-end X-ray crystallography, that can be built in only few places, these facilities can be built anywhere, and you have now, as a student, you have access to the same facility that you will have access to in best of the institutions in the world. So we are very excited with the prospects of structural biology in general in the country, and I'm more excited that our students will have a chance to work with these high-end facilities without even sort of depending on the facilities abroad. Just one or two slides I wanted to share quickly just for the students. So when I started all this back in 2014, some of you might have already heard about it. I have talked about it in multiple sort of lectures. It was in July 2014 when I was presenting my vision in front of a committee. And it was not at IIT Kanpur, it is in a different context somewhere else. And basically I presented what I want to do for the next 10 years or so. So essentially what we have done over the last 10 years or so. And one of the members of the committee, who is one of the most decorated scientists in the country, this award, this director, that director. And that person looked at me and said, and I quote, "Come on, who are you kidding? This cannot be done in India." Right to my face. And the committee had 17 members. A few of them were dozing off in the back. But even those people who were awake, nobody said a word. And then I looked at the person and said, "Thank you very much for your feedback. We are still going to try." And try we did. But the story doesn't end there. The reason why I'm sharing with you this story is that the same person over the years has become one of the biggest fans of our work and biggest supporter of our work. So if you believe in yourself, if you really dream big, if you have ambitions, you can even turn your critics into your supporters one day if you keep at it. So this is something to keep in mind. As I said, I've been back in India for 10 years. And the best part of my journey so far, in all honesty, has been to work with our wonderful students and fellows. They come from many different places. They have very different background. But I just cannot imagine that it would be possible to have the same kind of vibe and same kind of team spirit that our students have to have that elsewhere. So I've seen students in Europe, I've seen students in States. People mind their own business, and after their work is done, they leave, they go home, come back the next day. In our labs, what we have noticed that our students are very sort of closely knit in a way. They have really very, very high team spirit. They work with each other, they collaborate with each other. If one person is struggling, other people will come and provide support, and they will say, "Chal chai pi ke aate hain, chal coffee pi ke aate hain." They will go for tea breaks and coffee breaks, they'll discuss, and when they come back, you see that the person who was struggling before that coffee or tea break is now in better spirits, has better ideas, maybe looking at the challenges in a better way. And to be able to see that, this has been it has been a true blessing for me to see that in our students. Their collaborative spirit, their team spirit has no match. And I'm very, very thankful to our students, and I hope that the students here in the audience, you feel the same way and you work the same way. Science is a collaborative endeavor, and working together we can certainly achieve bigger things and greater things. So thank you very much, and I'll be happy to answer any questions.
Excellent talk.
Thank you.
And I mean, your journey is also motivational as motivational your work is. So I have a very basic question. I just wanted to know, when you design these drugs and you're checking them for the specificity for GPCRs, how do you know that they are not binding to any sort of other receptors? Like there are also, I mean, I know that you have not covered everything, but just wanted to know how we, like, because we are saying that we're trying to reduce the side effects. So how exactly that works?
That is a fantastic question. So in fact, when you try to develop a ligand or a drug for a specific receptor, you test it on other pool of receptors as well to make sure that it does not have any off-target effect. But obviously there are 20,000 genes in our body. It's not possible to test every drug or every drug-like molecule on the entire proteome. And that is often one of the reasons why some drugs have off-target effects. But in the context of GPCRs, we have the libraries of the entire GPCRome, and we can quickly screen and make sure that these compounds do not have any off-target effects. But it is very, very difficult to rule out that they will not touch any other target in the cellular context.
So, wonderful talk. I just have some simple questions. How difficult is it to purify and re-embed that in the membrane? And how much protein is required to proceed for cryo-EM?
So membrane proteins are believed to be more difficult than the soluble proteins because you have to take them out of their native environment, that is the biological membrane. But it is feasible, it is possible to do it. There are many, many labs across the globe who are doing it. Some proteins might be more difficult than the others, but it's not considered impossible anymore. In terms of the amount that is required, it all depends on the quality of the protein that you have. If you can imagine that, we are putting maybe only 10 micrograms of a protein on the cryo-EM grid. And if that is of high quality, you do not need to prepare the protein ever again. But to get that 10 microgram high quality protein, you have to do a lot of optimization. So the requirements are typically sub-milligram scale now.
Okay, and those nanodiscs, were they the longer one, larger one, or medium-sized?
So there are different types of nanodiscs. So you can make them in different sizes and different dimensions, just by playing with the lipid to protein ratio and so on. So if you're working on a receptor de novo, then it's primarily trial and error. But if you're working on a receptor for which some antagonists and some agonists are known, then of course you can go off of the known information and see, yes, based on the literature, based on the structural information that is available, and you can design the molecules in a targeted manner. Yes. So that is known as the concept of allosterism, and a lot of GPCRs, they have, or it is possible to design positive and negative allosteric modulators. And in many cases that are also being explored as potential drug-like molecules. So that is possible to design non-orthosteric site compounds which can then change the pharmacology of orthosteric compounds. Yes, that is possible.
I have one general question.
Yes.
Have you been successful so far, like getting an active compound or like a drug for any of these GPCRs after all this optimization? I mean, going through the structure optimization and all.
Right, so structure-guided drug design involves the ligand design first, and then when you say drug, it becomes a drug only after all the clinical phase approval, as you know. So we have been able to design new drugs with specific or desired functions encoded in them. Now they are in preclinical studies. Nothing has really gone through all the phase clinical trials, so nothing has become drug per se. But designing molecules that can activate or inactivate the receptor using the structural template has been possible.
And it is a very nice talk. So I'm working on a transcription factor and try to solve that structure and purify. It is a multifunctional transcription factor. So it has huge IDR, intrinsically disorder regions. So have you deal with any IDR proteins? This has a multifunctional role, and have you trouble any kind of the purification and all that?
Right. So these IDR containing proteins continue to be challenging, but there are ways to sort of address it. Obviously, if the regions which are disordered are not required for functions, you can always truncate them or delete them, or you can develop crystallography chaperones such as antibodies or nanobodies to stabilize them and lock them in specific conformation, and that can help you. So something like that, these kind of approaches have been used in the context of other IDR-containing proteins. Yes.
Good afternoon, sir. First of all, it's an honor to
Thank you.
be talking to you. I'm a MSc first semester student, so my question is not related to your work specifically. I have two questions. One is that since you just mentioned that you came to India 10 years back, I want to know when it comes to IP creation, countries like Germany, US, they are way ahead of us. I want to know what is the mindset difference from your standpoint? And my second question to you is that if you have to start all over again and you are given this opportunity that you can venture into this journey of entrepreneurship, I want to know what domain in biotechnology will you choose to solve the problems of healthcare, specifically in the context of India?
Thank you. Thank you very much. In terms of mindset, I believe that, earlier, for example, so I did my PhD abroad in Germany and then my postdoc in the United States. Even our students now, they go for, PhD and postdoc abroad, and that is completely fine. I believe back in the days, infrastructure used to be a serious issue where our infrastructure was not at the same level as in Europe or in US. Now I believe in many, many areas, I'm not saying that in every area we are at the same level, but in many, many different domains of science and technology, we are at the same level. You have access to the same plate reader, you have access to the same genomics platform, you have access to the same proteomics platform. Cryo-EM example, as I said, you are using the same machine here that you'll use at any other institution in the world. So these things are changing, of course. So we are improving in terms of infrastructure to get at that level. In terms of mindset, I don't really see a huge difference other than you have to be passionate about what science you are doing, and you really have to have high ambitions. And even when people tell you that it is not possible, you have to show them that it is possible. So you should have that fire and that sort of urge to really do something good inside you. In terms of entrepreneurship, this is something which will keep changing. There, problems which are, for example, more prominent right now, 10 years later, it could be other problems. As you know, a couple of years back, everybody got into COVID because of the pandemic. But even in healthcare, for example, these different pathogen-host interaction to combat the pathogens' antimicrobial resistance, or even the metabolic disorders which are looming to be pandemics, these could be very, very sort of promising directions if you want to take. These things take time, as you know. So designing a drug, designing vaccines, it's not an overnight thing. It takes years and years and years of work. So you have to be prepared for that. But any human disease, for example, is fashionable now, will remain fashionable. The context, the kind of disease might change or evolve over time, but this is something which will always remain of societal relevance. That is human disease, how you can cure it, how you can design new drugs or new vaccines and so on. Just to give you one example, there will be other areas as well. Yes.
Good afternoon, sir. It was a privilege to listen to your talk. So first, I have two questions, and the first is related to what you have talked about. Sir, as you talked about that GPCR and interaction of ligand, and you said that it might induce two pathways, either GPCR or beta-arrestin pathway. So I want to know that is it just ligand oriented, or is it, or you have also worked upon the outer environment, which can affect either of the pathways, or is it just sort of a probability, like a lot of GPCR pathways which might induce the beta-arrestin pathway maybe. So have you also worked on it? And if yes, please enlighten us with that.
No, thank you very much. That is a very pertinent question, very good question. This was a little bit of oversimplification, so to say. Obviously, these receptors can have additional pathways, additional crosstalks with other signaling molecules, other signaling receptors. Even in the native context, even our body can produce different ligands that can be biased on a given receptors. So it's not that only synthetic ligands can be biased, even natural ligands can have biased propensity. It is also possible to have biased receptors in our genome naturally. In fact, we have discovered that there are several receptors, chemokine receptors in particular, which are natural receptors in our immune cells. When they bind to their natural chemokine ligands, they will selectively activate beta-arrestins but not G proteins. So ligands will have the major sort of say in directing which pathway is going to be activated. It is possible to do it in vitro, but nature has also done it. And the mechanism is that different ligands will induce different conformation. By conformation, I mean different structures or structural changes in the receptor. If it is structural change 1, that is more sort of permissible for G protein binding. If it is a different conformation or structure 2, that will be more permissible to beta-arrestin binding. And that is the mechanism which drives these differential signaling.
Okay, so one more question. Like I've talked to some of the scientists from either Pune, and they have this take that India is a lot more, states and other countries are a lot more ahead than India, almost 40 years ahead than India. So what's your take on it? Like, is it really, do you face any challenges being in India as a scientist and a researcher?
Well, that's a very good question. Do I dare say that it's a rosy bed, everything is hunky-dory? Absolutely no. There are many, many hurdles. But it is our responsibility to try to do cutting-edge science and world-class science despite those hurdles, and at the same time, try to improve the situation for the next generation. If we are not going to do it, then who is going to do it for us? This is our motherland, and we have to deal with the hurdles. We have to overcome them. We have to have that fire despite the problems and the challenges that we are still going to do good science. And then in parallel, we have to do our bit in terms of improving the system at whatever level. So, for example, whatever little I have been able to contribute in terms of the cryo-EM infrastructure across country, it's not that it's only going to benefit my lab or our students. It's for everybody. These are all national facilities. As I said, this cannot be an individual effort. This has been a collective, collaborative effort of many, many people, funding agencies, and so on. But together we have been able to do it a little bit. And similarly, I think in other fields, we need to do the same, in other domains of research. I believe, and I honestly believe, that we have real advantage, that our students are absolutely phenomenal. And if you guys maintain your motivation, if you are ambitious, there is no reason why you cannot perform at the same level as your counterparts abroad. Now, only dream that I have left is that one of these days, one of you will get a call from Stockholm, and I will be around to cheer for you. That is my only dream that I want to see getting fulfilling in my lifetime. And I honestly believe that it's going to happen. One of you is going to do it. And when you get that call, do not forget me.
Thank you so much. So this is one of the most important reasons why I follow you a lot, and I really admire your work.
Good afternoon, sir. Sir, actually, my question was, you described to us the analgesic binding of drugs like morphine with the GPCR pathways, specifically on G protein and beta-arrestin pathway. So in case of development of a drug which has less side effects, or might I say none at all, is the beta-arrestin pathway being blocked or completely shut off, or it's not just binding? Like, what is the mechanism? Can you kindly highlight on that?
Yes, it's a very good question. I did not go into that technical details, but at least for morphine, the mu-opioid receptor system, the way it works is that the G protein bias ligand will put the receptor in a conformation or in a structure that it will not get phosphorylated, and therefore it cannot bind beta-arrestins. So even in the cellular context, in the physiological context, beta-arrestins are there, the receptor is active, the G proteins are active, but beta-arrestins will not be able to interact or bind with the receptor, and hence beta-arrestin pathway will not be activated.
I think that was a fantastic interaction and where students got inspired by your lecture. I invite our respected Lakhotia sir to come and felicitate the speaker today. So despite all, you have engagement in classes, research and all, you turned up pretty well. So thank you for that. I ask now our Professor Madhu Tapadia madam to deliver a vote of thanks to the speaker and the audience.
Good afternoon everyone, and thank you Dr. Samarendra for inviting me to present the vote of thanks. And I think it's a privilege for me to thank today's talk, the speaker, the organizers and the audience for a very fantastic talk. And I first of all thank Dr. Arun Shukla for taking time out of his busy schedule. Though he said that his students do the work and he spends time drinking coffee, but I'm sure in between the interpretation of data, that's one of the most important aspects of meaningful science, he does it. So it was very modest of him. And one of the best things, I'm sure the students also will agree, that the way he built his talk from very simple to going into the very complex aspects of it, still not so complex for us to understand what exactly he's doing. And he has explained very nicely the intricacies of how designing a drug, how can a drug be designed. And then, though it all seemed very simple, but I'm sure there must be lot of difficult hours before getting the achievable target. So I really thank you for agreeing to come over here and speaking to the students and the audience. I also thank the Infosys committee for organizing this lecture at Institute of Science, Banaras Hindu University. And as we know that Banaras Hindu University was the vision of Pandit Madan Mohan Malviya ji, and he aspired to translate very simple science. His vision was like from very Prachi and Pratichi, old and new amalgamation. So this is very nicely, we have seen it today. I thank all the senior teachers of Institute of Science who are present over here and for this lecture, and their presence means a lot to us as well as the students. I thank our director, Professor Sanjay Kumar sir, for organizing this lecture and making the hall and everything available to us, and our dean, Professor Sukhmender Singh sir, for again organizing this lecture for the benefit of the students. And seeing the crowd of the students, I'm sure it was it was successful. And I thank all the faculty members and all the students who are present over here for making this event a very successful one. Thank you so much.